Yeah, I see it. YouTube on here.
Oh, and Adrian, when you give your talk, if you want to let people know how they can see your second talk that's later today, feel free to give that a plug.
Oh, sorry, I was muted. Yes, yeah. So I have a sort of small reminder, and yeah, so people can ask me.
I was really impressed when I saw you were planning on giving 2 talks in one day, so good for you.
Thanks. I was sort of unplanned because when we talk, it was like, yeah, whatever. End of July seems fine. End of June seems fine. And then I signed up for this other one, and I thought it was gonna be at a different date. And then they told me they chose me for today's date, and I said, oh, well, yeah.
You're like, okay, I guess that'll be my entire day.
It's better than none. And they're 2 different talks, so yeah. So it's not like I can reuse it or.
That's what I would have done. I definitely would have given the same talk twice, but you prepared too, and that's awesome. I have to go into lab as soon as this is over, but I will try to catch it. Do you know if they show those talks live or not? Or sorry, after the fact? Or is it just
so?
live?
Yeah, I think they're also recorded, and you can look at them up. Yeah.
Okay, great. That's really awesome.
Yeah, I haven't been able to see many because, yeah, as you might imagine, I don't have a lot of free time, but I think so.
The timing's really, really tricky. But some of these live seminars like ours, you know, trying to make things available online, I hope really helps people out who
Yeah,
have
it's much better,
complicated
yeah.
schedules. Yeah, that's the goal. I'm looking at this favorite lab equipment, and I think ice machine is really underappreciated. It's really essential. It's a really good point.
Hello. Hi, Jen.
Hey, Jim, how's it going?
How do I get rid of this thing of what's your favorite lab equipment?
You don't. We haven't started yet. But if you want to vote, I know it's been a while since you've been in the lab, but you can go to that website and enter in the code, and then you get to type in your favorite lab equipment. But we'll take that away in just a minute as soon as we get started.
Great, good, thank you.
Just usually we have a lot of people trickling in just a couple minutes into the hour, so but we'll get started in just a minute now. All right, I think let's go ahead and get started. If Adrian, if you want to go ahead and start sharing your screen. So hi, everybody. Welcome to the 8th installment of the Cell Migration Seminars series. Before we get started, just a quick reminder about questions. So we'll do them all at the end, and you can type your question into the chat window, or alternatively, if you want to ask your own question, which we encourage to make things a little bit more interactive, just type in question and we'll call on you and you can unmute your microphone. Okay, so this week we're going to have 2 talks. And so the first one is by Adrian Jacobo. So he has a background in physics and he's now studying development and regeneration in the Hudspeth lab at Rockefeller University. Excuse me. So Adrian studies 2 mechanosensory organs, the inner ear of mammals and the lateral line of zebrafish. And using these organisms, he works on several problems of pattern formation and self-organization using a combination of experimental and theoretical tools. And so we're really excited to have him kick off our talks today, and I'll hand the microphone over to you, Adrian.
Thank you, Jennifer, and thanks to the organizers for putting together this fantastic seminar series and for choosing my work to present. So as Jennifer was saying, I work on the zebrafish lateral line. So let me tell you a little bit about the system to start. So this is a schematic of a 2-day-old to 3-day-old zebrafish larvae. And these dots here are what are called neuromasts. They form part of the lateral line. So this is a side view of one of these organs. And these are mechanoreceptor organs. So the fish use them to detect water current. So they detect the velocity at which they're swimming in the water and also the presence of predators or prey in the surrounding fluid. And they do this thanks to these specialized cells called hair cells, which are mechanoreceptors. And they are mechanoreceptors because they have these organelles called hair bundles, which are formed by actin protrusions and the primary cilium of the cell called the kinocilium. So this long cilium here. And these protrusions, so these hair bundles extend outside of the skin of the fish, where they are in contact with the fluid. When movements of the fluid deflect the hair bundles, there's mechanotransduction channels on the bundles that are open. Then ions enter the cell depolarizes, and then this relays information to the brain about the direction of the water movement. And they have directional sensitivity because there's 2 different populations of hair cells. So one is these cells that I mark here in orange, or here in this top view, which are sensitive to water movements from the head of the fish towards the tail. And there's another population which is sensitive to the opposite. So these cells marked in blue are sensitive to movements of water from the tail towards the head. And here you can see, so these black dots mark the position of the primary cilium, which is what you can see here in this picture. So this is a confocal image of cells where actin was labeled with GFP. So because we label actin, the primary cilium of the cell, which is tubulin based, is unlabeled. So you see it in black, but the position of the kinocilium marks the direction of sensitivity of the cell. So what I want you to notice is 2 things. So one is that this organization of the hair cells is symmetric. So cells of opposite polarities are facing each other. So you can see it better in this schematic. So all the cells of one polarity are on one side of the neuromast. The cells of the opposite polarity are on the opposite side. And the other thing is that here we're labeling only the hair cells, but there's a whole sea of other cells called support cells that are surrounding them which are unlabeled here, and they're sort of important for what follows. And so what we want to understand is how this mirror symmetric organization is achieved during the development of the organ, and how it's maintained when the organ regenerates. So one thing that the zebrafish can do that we can't is to regenerate. So these hair cells are also present in our inner ears, and they're responsible for your senses of hearing and balance. So you're hearing what I'm saying because you have hair cells in your inner ear transducing the vibrations of my voice into electrical impulses. But if you lose your hair cells, they never come back. And in the case of the fish, they are able to regenerate their hair cells, and every time they regenerate, they're able to reconstitute this mirror symmetric organization. So, so this is the question that we want to understand. And to start to understand this, first we need to see, look at how these cells develop. So the cells start, a hair cell starts from a progenitor support cell. This support cell will divide into two, and then they will undergo a process of lateral inhibition, about which I'm going to talk a little bit more in detail in the next slide. But for now, what you need to know is that this process breaks the initial symmetry and gives the cells two distinct biochemical identities that I'm just schematizing here with two different colors. And then the cell that has the orange identity will end up becoming one of the polarities, which we call now the minus polarity. And the opposite cell, the blue cell, will become the opposite polarity, which we call the plus polarity. So they do this. So this lateral inhibition process is mediated by notch. And this is, I just give you the short gist about it, but I'm gonna give a talk later today at the Stower seminar series, so or you can also read this reference. But if you want more details about how we know that this is the way the process works, so you can send me an email or a message, and I can give you the details if you want to join at the seminar. But what is important to know now is that the cells undergo a tug of war in which each cell tries to make the other one produce less notch while producing more notch itself. So this creates a winner-takes-all situation in which both cells start being symmetric and having similar quantities of notch, but then one wins and suppresses the other. And then, depending on the amount of notch, there is an activation threshold. So the cell that wins will adopt one of the polarities. The cell that loses will adopt the opposite polarity. Okay, so this is a way that the system makes sure that you have two cells of opposite polarity coming out from a symmetric cell division. And so what happens is that this process can give rise to two possibilities. One in which the negative cell is the one more anterior, so it would be to the left of your screen. So the orange cell is to the left of your screen, and in that case, the bundles are facing each other. Or you can, with equal probability, give rise to a situation in which the blue cell is the one to the left, or the more anterior, and therefore the bundles will look away from each other. But when we look at the organs, what we see is that we never observe this configuration. We always observe a configuration in which the bundles are facing each other. Okay, so the question is, how does this happen? So how is the organ suppressing one of the states and only allowing us to see the other one? So to answer this question, first we need to look at how the cells develop a little bit more in detail. So what you're going to see here, it's a movie of two cells that just divided, and you're going to see that when they divide, they separate from each other and they extend protrusions in opposite directions. So this cell is extending protrusions to the right, this cell is extending protrusions to the left here. Okay, so these protrusions are correlated to, so these protrusions appear early during the life of the cell, and then when the cell matures and forms a bundle, the direction of the protrusions is correlated with the direction of the bundle. Okay, so this suggests that the biochemical identity of the cells changes the polarity of the cytoskeleton in a way that influences both the protrusions and the direction of the hair bundle later during the life of the cell. And we know that this is more than a correlation because in the case of cells that lack notch, so as I told you before, cells with high notch will adopt the blue polarity, so the plus polarity. Cells with low notch will adopt the minus polarity. So cells that have notch mutants that have no notch at all, all the cells adopt the same polarity and have protrusions. So you're going to see a pair of cells here that have both protrusions towards the left of your screen and have the same polarity. And the bundles of these cells are all oriented in the same direction. Okay, so there's a link between, again, the polarity of the bundle and the polarity of the protrusions. And these protrusions have the effect that they drive cell movements. So in the case of the wild types, because the protrusions point away from each other, you get into a situation where the cells move away. So the cell distance increases with time. And in the case of the mutants, because the protrusions are pointing in the same direction, both cells move in the same direction, so the cell distance remains constant. Okay. But if you look at the translated distance of the pair, so if you look at the position of the center of mass and how that changes with time, you see that in the mutants, the translated distance increases with time, while the in the wild types, because both cells are moving away from each other more or less at the same speed, the center of mass stays constant. It doesn't move. Okay, so to try to understand how this process works, we don't have a lot of understanding of the molecular mechanisms driving these processes. So what we did is to write a coarse-grained physical theory, so what is called an effective theory, based on the physics of soap bubbles, and that you can adapt to now a cell, which is not a simple soap bubble, so it can actively change its physical properties by spending energy. And so this physical theory describes the surface tension of the cells. So you have both cells of the pair, and so each cell has a surface tension associated to this part of the interface, which is the interface between the hair cell and the surrounding support cells. And there's another energy associated to the interface between the two hair cells. Okay, so we call the surface tension of these, we call it gamma S, and the surface tension of the hair cell interface, we call it gamma H. And what this tells us is that, and we can write the total energy of the system as just the sum over the areas of all the energies. So this is for the more mathematically inclined people in the audience, and you can look at the details in the paper, or you can ask me later. But what is important to understand from this is that this formalism, what it tells us is that for the cells to have protrusions, the surface tension across the hair cell to support cell interface has to change with space. So it can't be homogeneous. It can't be always the same. So what we assume, the simplest assumption, is that there is a linear decrease in surface tension from the hair cell interface towards the other side of the cell. And so this is how it would look like. And we call the total difference between both ends of the cell, the total difference in surface tension, we call it delta gamma S. And as a function of this quantity and the baseline, so what is the ratio between these two quantities, gamma S and gamma H, we can reproduce all the different stages that the cells go through throughout their maturation life. Okay, so initially they start with a high ratio for the surface tension between hair cell at the hair cell interface with respect to the hair cell to support cell interface. So they like to stay attached and form a very flat and large interface between the hair cells. And on the other side, they want to repel the support cells, so they form a spherical interface. And then as the gradient starts developing, the cells start to move, okay? And I'm going to talk a little bit more about this stage in the next slide, but they start to move. In principle, they can move away from each other. Then they reach this point where the surface tension is enough that they want to increase their area in the hair cell to support cell interface. So they start forming these protrusions that I showed you before. And then finally, they reach the point where energetically it's more favorable for the cells to form a complete hair cell to support cell interface than to keep the hair cell interface. So the hair cells separate from each other, and then there's a support cell that wedges in between the two. Okay, so this framework allows us to explain all the different stages that we see in sort of a parsimonious way. But it also has another implication that is related to this region, so this part of the developmental process, which is that these gradients are coupled to the internal biochemical state of the cell. So in this case, where the minus cell is on the anterior side or to your left, the gradients are facing each other. So the high part of the gradient is facing each other, which means that the cells want to move away from each other. Okay, so the cells want to move down gradient because that's where they spend less energy. So they want to move down gradient and they move away from each other and they separate. But in the other case, where the blue cell is the one that got selected to be to the left, the gradients are facing away from each other, which creates a movement of the cells towards each other. But because the cells are attached at the interface, this creates a rotation. So the cells tumble one with the other. So they exchange their positions and tumble. So, and then they reach this configuration where they are free to move away from each other. And you're wondering whether this is just the theory or the cells actually do this. And this is a movie of this process happening. So the cells do this. So you'll see a cell pair divide here, and then you'll see the interface form and the cells flip their positions and then move away from each other. Okay, and they do this 50% of the time. So this is compatible with this idea that the cells undergo this unbiased lateral inhibition process. And then if they get it wrong, if the blue cells or the positive cell end up on the anterior side of the neural mass, then this mechanical process just rearranges them and they end up into this situation in which the negative cell end up being to the left. And in the wild type, they do this 50% of the time. In the notch mutants, because all the cells are the same polarity, they never do these rearrangements. So with using our mathematical theory, we can also reproduce the kinematics of the rotation, so the speed of rotation as a function of the angle. And so this is the theory, the black line, and the dots are the experiment. Just to summarize, what I expect that I show you is that the symmetric organization of the organ is achieved by two steps: a biochemical step that creates two possible states, and then the mechanical step that maps this state into the state that we always observe, with the minus cell always being the more anterior one. And with that, I would like to thank Jim, my mentor, and Anna, who was my collaborator, who did half of the work in this paper, and Nick, who helped us with the fish. And I would like to thank you for your attention. I'll be answering some questions now.
All right, thank you, Adrian. If people want to unmute their mics and clap. Just want to give you a sense like you're in a real auditorium. So we'll go ahead and take some questions, and guys, feel free to ask them yourselves or post them in the chat. So the first one is from Ankita Jha, who asks, what's the role of adhesive forces in the rotation of the cells?
So we don't know. This is something we would really love to know. So our theoretical framework, it's just, as I was saying, it's just a coarse grain description. So that includes adhesion forces, cytoskeletal changes, passive changes of the physical properties of the cell. So adhesion forces are part of this effective surface tension, but we don't know exactly how. Yeah. So if I had to guess, I would say that probably there are some adhesion proteins that have a gradient across the cell, and this is what is part of what is driving these gradients. But right now we don't know.
I have a question. I'm sorry if I missed this because I'm trying to manage a couple things while moderating, but I was wondering, do you understand what stops the rotation from happening?
Yes. So after the cells rotate, their gradients are pulling away from each other. So the cells just move away. Okay. So one thing that is underlying all of this is that the cells do have a sense of the anterior posterior axis. Okay. And their biochemical identity gives them sort of a sign with respect to this axis. So one of the cells wants to move in the anterior direction, the other cell likes to move in the posterior direction, essentially. Okay, so after they rotated, because the cells are now in sort of their natural positions, they just move away from each other and they keep moving away.
Okay, great. Thank
We've
you.
got a question from Rashmi Priya.
Hi, Adrian. Terrific talk. I have a question, and maybe I missed it, but like you say that it's a stochastic process. So is notch pre-patterned there? Do you already see notch less or more, or does it like gets amplified later on, eventually when these cells are moving and undergoing rotations?
No, so this,
Yeah.
and actually I'm going to talk more in detail about this in the seminar this afternoon, or depending on where you are tonight. So we know that the initial state of the cells is completely unbiased, and the amplification is just of stochastic noises in the cells. Yeah. So there's no pre-patterning of the cell division.
And just one quick thing. So why do you see less protrusions in notch mutants? Do you have any idea why?
We don't see less protrusions. We see the same amount. All the cells extend protrusions, but they extend them all in the same direction.
Okay. Thank you.
Okay, thanks. We have a question from Amrinder Nain, who says, nice talk. And is there a chirality to rotation? I believe you only show clockwise rotation. Is it driven by the positive cell?
No, so there's no chirality in the rotations as far as we know. It's a tricky question to answer because there might be some slight chirality that would need us to acquire a lot of data and follow a lot of these rotations. But as far as we know, we see them rotate in both directions. We just, in the plot, we just normalize to, so we do everything modulo pi so that we can overlap all the curves, but actually they can rotate one direction or the other.
Last question for Rickard.
Yes. Hi, Adrian. Thanks for the talk. So I'm wondering about contact inhibition of locomotion between these cells. Does this have any effect? Because I would think that since what you're saying is driven by the polarization of these cells, if they repolarize faster than they rotate, maybe they would be able to detach before rotating. I don't know if that's possible. Essentially, what's the role and timescale of contact inhibition of locomotion?
Yeah, so we don't know for sure. So according to our theory, the separation of the cells has to happen after the rotations, because the cells have to so they have to achieve this threshold of being more energetically favorable to attach to support cells than to hurt cells, and this is what drives the separation, and to achieve that they have to go through the step where first they move away or they rotate. Okay, so but there could be something that we're missing. I'm not entirely sure how we would test it experimentally.
Okay, thanks.
Okay, thanks. Thanks, everybody. We actually have to move on now in the name of time, but if people want to have additional questions or continue the discussion, please do so on the Slack channel. All right, so Alessandra, if you want to go ahead and start sharing your screen. Great. I'm delighted to introduce Professor Alessandra Cambi. So when I was looking through her bio, I was really impressed and surprised to see that she actually has received 2 PhDs, the first one in biology and then a second one in medical sciences. So I can't believe you managed to get through 2 of those. So, so she's currently a professor of cell biology and the head of the Department of Cell Biology at Radboud University Medical Center. So she currently leads the nanoscale cell biology group, where they study fundamental mechanisms of leukocyte biology in health and disease, and they exploit various high-resolution microscopy techniques and quantitative biophysical approaches. And so based on the movie we're seeing now, I'm expecting we'll get to see some of those very cool high-resolution microscopy techniques. So
that's it. Thank you very much, Jennifer.
Thank you.
Thank you. So it's of course a pleasure also for me to follow up on this very interesting talk, and I'm always very envious when people can make this beautiful modeling that supports so nicely cell biology. We don't. So if you feel after this talk that you can contribute, please just send me a message. So thank you, of course, to Jennifer, Adam, and Becky for organizing this seminar series. There's been really a light entertainment for me every week to follow in the past 8 weeks, I realize. And so today is my turn to share with you our interest in the mechanobiology of dendritic cells And this, what you see here crawling around, is a dendritic cells, a human dendritic cell. So dendritic cells are the sentinels of the immune system. They come from precursors. Then they will usually colonize the peripheral tissues in a sort of immature state where they will patrol for antigens. And when the antigens are encountered, like pathogens, the dendritic cells are getting activated, and they undergo a real strong phenotypical change. They will become what we call mature, highly migratory, and they will start processing the antigen and really move to the draining lymph nodes, where they will present antigen-derived peptides to the T cells. Thereby, they do initiate the immune response, and that's why we call them also very important tools for immunotherapy strategies. And that's not surprising that, in fact, dendritic cell-based vaccines have been used as an anti-cancer immunotherapy. The contribution of my lab in the past years has been to increase our basic knowledge of these cell types, basic cellular and molecular mechanisms that regulate their function. And one of the things that started to interest us in the past years is, of course, the contribution of mechanical forces. Now, we know, of course, that all organs have different pathophysiological degrees of stiffness. Usually, you have dendritic cells that are in the soft range of tissues in here. But of course, tissues also undergo all kinds of changes, right? So we know that tumors, in some cases, solid tumors have this stiffness increase. We also know, of course, that you have fibrotic tissue and formation of damage and scar tissue. And of course, also implants, and immune cells are actually the first cells that colonize also implants. And so this is, of course, another way of sensing different stiffnesses. Despite dendritic cells being always in this environment, we still culture them here. And so we thought, well, maybe we could just start to understand whether there is some kind of optimum function for dendritic cells in some kind of stiffness environment. So we know, of course, that the typical, basic fundamental seminal studies that showing a stem cell substrate stiffness can affect stem cells. The stiff substrate will lead to osteogenesis, the soft substrate to adipogenesis. We all know, of course, studies from extremely excellent labs also showing cardiomyocyte activity. Cardiomyocytes beat the best at a certain stiffness, and then in a scar-like rigidity, they start to beat much less. And so our question was basically, so what about these monocytes and macrophages and dendritic cells that go around continuously in our body? What do they feel? And is that any important at all? So we call it our mechanobiology adventure. And the reason is, besides that it sounds funny, that we tackle from 2 points of view, from a more mechanobiology point of view, a cellular level. So those substrate stiffness affect dendritic cell phenotype and functions. And the second question that we have is, do we have at the more nanoscale level, do we have a remodeling of dendritic cell cytoskeleton in response to substrate mechanical properties? And so starting with the first question, I can tell you that we can, of course, isolate human monocytes from the blood. And then we start culturing them on soft wells substrate. And these substrates are of different stiffness. So this is considered pretty soft. And then you go up to 12 kilopascal, which is already a little bit stiffer. And then we use this 50 kilopascal as well. We differentiate the monocytes for 4 days, and then you get what we call the immature disease. And then if you give a maturation cocktail, which is again very well established in the field, then you get the harvest mature dendritic cells. And the immature disease are in the tissues, and the mature disease should be going to the lymph nodes. So the first thing that we noticed on immature dendritic cell is that at 12 kilopascal, then we had this decreased expression levels of total beta-2 integrins, whereas the beta-1 integrins will stay the same. It's not that they disappear, of course, but it's much less. And because we were afraid that this would be something funny going on with this 12 kilopascal batch of substrate, you can see we tested many, many different donors just to be sure it was not something funny, and also different batches. There was not something funny with the substrate. The consequences of having lower beta-2 integrins at this stiffness value was that this disease were less able to form protrusions that are called podosomes, about which I will talk about later on in the second part of this talk. And the main driver of podosome formation in dendritic cells are beta-2 integrins, although beta-1 are also involved. So it's not, again, that they are completely gone, but there is much less formation at 12 kilopascal with respect to the soft tissue-like stiffness. We've also seen that the 12 kilopascal have also a negative effect on the expression level of receptors that are involved in pathogen recognition, which are obviously receptors that are very important for immature DC activity. And as you can see here, the expression level of two of them, the mannose receptor and DC sign, is much less, is much lower on the 12 kilopascal, which results also in a lower rate of albumin internalization, and you see it here. The good thing is that this is not just an overall not feeling well of the cells at 12 kilopascal, because if you give them transferring the internalization through clathrin-coated bead will just be very fine. So there must be something specific that is connected to this, to the presence of these receptors. And then we moved to the mature disease, and I give you here just a few, an example of what we've looked at. Of course, mature disease in terms of migration are known to use their CCR7 receptor to follow the gradients of CCR21 chemokines and reach to the lymph nodes. I think there's beautiful work from Michael Sick's lab, so I don't think I need to introduce this system to this audience. But what we also saw here again was that in many donors, the expression levels of CCR7 were decreased. And in fact, also the transwell migration was decreased, and the transwell towards a CCR21 reservoir. So then we wonder a little bit in the literature, as like, okay, what's this 12 kilopascal doing? And we understand from work from other labs that this 12 kilopascal is somehow considered a sort of cutoff value, for instance, for stage 4 liver fibrosis. In the lungs, we have learned that you have a healthy lungs of around 2 kilopascal, but a fibrotic lung, it's about 17 kilopascal. And there's a seminal review of Paul Jenny and Miller already it's 9 years ago, but I think it contains quite a lot of interesting information, where it says that 12 kilopascal or greater can in fact lead to an aberrant cell cycle progression, so that the tissue overall can be abnormal. And so the cell, the immune cell that might be finding themselves in those tissues could also feel something weird. So I can tell you that this is to be continued, because we think might be very interesting to understand the role of the dendritic cells in, for instance, a fibrotic environment. Another, what about the mechanobiology? So what about the nanoscale? So what happens when disease feel the substrate stiffness? How do they respond? What are the structures? So to do this, I need to introduce you to the beautiful world of invadosomes. And invadosomes are it's the name for protrusions produced by made by many different cells. Actually, invadosomes is a collective name for two different protrusions: the good guys, the podosomes, and the bad guys, the invadopodia, which are made by cancer cells. Many cells make these protrusions. Myoblasts use them to fuse endothelial cells. A very nice lab work from the Genot lab about blood vessel sprouting. Osteoclasts also use them to digest the bone. The anchor cell, beautiful work of the Sherwood lab in vivo, C. elegans development. You really see the anchor cells producing all these small invadopodia, trying to poke in between other cells and continue their migration. Leukocytes, of course, it's a big group of people interested in this. And we've seen, for instance, possibly the leukocytes using this protrusion to, again, feel the substrate and finding, for instance, an opening to transmigrate during the transendothelial migration, and also trying to feel the surface of an endothelial layer and try to find a place where they can just protrude towards the gut ways or the airways. And of course, again, the cancer cells. So they will mimic the podosomes, and then you will see this digestion of the matrix, and the invadopodia have a very important role in a cancer dissemination and colonization of the healthy tissue. Regarding podosomes on dendritic cells, you see here that there are several functions that have been identified through the years, and not only by us, but also by other people working in the field. First of all, we notice that there is a topography sensing function. So if you put dendritic cells on some kind of substrate with topography, you see this beautiful alignment of these structures. If you allow the cells to make podosomes and on a substrate like a filter that allows the protrusion to proceed, then you will have some kind of antigen sampling machinery that will go towards some kind of antigen and be able to mediate antigen pickup. And this is actually also what is reminiscent of what happens here in this beautiful movie of the Ron Germain's lab, showing dendritic cells that are basically waving into the lymphatic sinuses, trying to capture these yellow particles. And we have also seen that, of course, podosomes are involved in this migration of these dendritic cells. This is an immature disease. They move. They're not fast, but they do move, of course, around. And they nicely colocalize with areas where there is substrate degradation, as you can see here in this gelatin-coated plate. And what we've seen in the past is that the moment that you sort of stimulate dendritic cells, immature dendritic cells, with the typical maturation agents, which could be prostaglandin E2 or LPS or TNF-alpha, you have an immediate dissolution of podosome. And that will be for us, we call them a sort of first step towards dendritic cell maturation, at least in an in vitro setting. So if you look now at this podosome that always are formed in nice clusters, and this is one portion of a dendritic cells, which is transfected with LifeAct GFP, and an image by Eriscan, I think you notice at least two different type of dynamics. In the past, we have called this the fit stomping at the level of an individual podosome. So you see one podosome growing like in intensity. That means that actin is being polymerized and it grows and it grows and it grows. And then it will start shrinking. So actin is depolymerizing. And by doing this, you have this sort of pulsating behavior of individual podosomes. But you also see that they are actually nicely connected by many filaments that are, of course, actin filaments. And so we ended up by finding that these filaments connect and mediate some kind of mesoscale organization of these podosomes in these small clusters, helping redistribution of components, but also redistribution of forces when the cells are actually exploring the substrate. Now, how does an individual podosome build up its pushing forces? If it is just a tower of actin, how can this actin find a mechanical force to push back towards the substrate and make a protrusion? And the second question we had was, so what do podosomes do when you put the cells on substrates that have a different stiffness? Do they react and do they change in their composition? How plastic is this system? Now, if you zoom in on one podosome, then you can distinguish two structures. At least in the field, we have this core and ring dogma, meaning that you have a core, a central enrichment of actin, surrounded by many proteins like vinculin and all kinds of scaffolds proteins. And so you have, so this will be the two different channels. Now, some of you might have seen this image before, and last year actually it was pretty famous, but these are two completely different things. It's a different galaxy. That's a different galaxy far away. Now, the bad thing of Zoom is that you really don't know whether a joke had an effect or not on your audience, which is muted and blinded. But nevertheless, I will proceed, assuming you like the joke and the analogy. So the point I want to make is that the core is composed by many different components, actin and also a lot of different actin binding proteins. And of course, the ring is also formed by actins, but also integrins, and as I said, vinculin and all kinds of other beautiful scaffolds or myosin 2A and formins. What we noticed was that actually in the core, we had molecules like alpha-actinin and WASP, cortactin and ARF23, which we know bind to actin, but seem to have a different preference depending on the actin structure. So actin and its interactors are in fact a very multifaceted population of components So if you look at these branched actin, then there are nucleators that prefer the branched actins. And there are nucleators that actually prefer more the bundles, whether they are parallel or anti-parallel. And then as you can see here, we have identified this cortactin and this alpha-actinin that actually within the core binds to two different structure of the actin. And so the first question that came up to our mind was, could it be that we have different actin arrangements within this 500 nanometer small podosome core? And so of course we started to stain for interactors of branched actin, and we found them like wasp, much more enriched at the core center. So if you do a profile and like a sort of section of many different podosomes, you find that the wasp has a much smaller full width half max of the peak. So it's always much more in the center of the core. Whereas alpha-actinin, which is preferentially interacting with bundles of actin, has this nice almost donut shape profile with respect to the actin intensity. And so if you then take a lot of images of more than 200 individual podosomes, and then you do the cross sections and you average them out, and you use vinculin as a sort of benchmark, then we know that we are looking at podosomes that are vinculin positive and actin positive. Then I think you would agree with me that the alpha-actinin gives this nice, almost roof-like staining with respect to the rest of the podosome, like a little cage all around the actin signal. Then we also discovered that, in fact, you all know probably that we have actin isoforms in the cells. What we didn't know a couple of years ago was that these actin isoforms seem to have preferences also for the type of actin structure in which they can be enriched. And so the beta actin isoform seems to have a preference for the branched actin arrangement, whereas the gamma actin isoform seems to be predominantly found in more bundle-like actin. And this is a work that has been, so this is information, is actually based on a beautiful work of the Kashina lab in the past years. And the funny thing is that these isoforms are, of course, very similar, but then they have this few amino acids difference at the end terminus that seems to make the difference. So what we did is basically starting to stain for the beta actin, of course, and the gamma actin using specific antibodies. And we have looked with two different techniques like Eriscan and Storm. And I think as you can see here, there is a gamma actin going from the bottom of one individual podosome up to about 700 nanometers inside the cell. And then I think you would agree with me again that the gamma actin is making this nice ring all around the much more enriched beta actin in the middle. And the same is actually confirmed also by the Storm images. So basically, if you do again an average of more than 200 individual podosomes and cross sections, you see again that the gamma actin has this roof-like or cage-like organization with respect to the beta actin. And so then we went back and we said, okay, that's very interesting. So the core seems to have two different things, something inside that is more branched and something outside it is more bundle-like. So what about the so-called ring area of the podosomes? So then I want to focus here on two components, well, of course, actin, but also especially myosin 2A, so non-muscle myosin. And so if you look at the dendritic cells, both fixed or living, and you look for actin at these clusters of podosomes, I mean, you really see that they are beautifully complex. They have all these connections, all these small filaments connecting the podosomes to each other, but also the podosome to the membrane. And then this is not an artifact of a fixation, but of course you also see it here also in living dendritic cells. And so then we knew in the past that non-muscle myosin 2A is important for podosome activity. So if you block myosin 2A with these inhibitors, you don't see anymore these oscillations, these vertical oscillations of polymerization and depolymerization of the individual podosomes. If you block myosin, you also lose the so-called mesoscale organization, so they do not fill the neighbors anymore. And after many attempts to transfect these primary human cells, we managed to transfect the myosin light chain fused to GFP, overexpress it. And then you see that very often you can see some intensity, some particles of myosin co-localizing with some of the filaments that are radiating from these different podosomes, but not all of them. And then we wondered, like, okay, so is there any organization, excuse me, is there any organization in this interpodosome actin network? And we found out that there is actually like two floors of this actin network. We have two vertical components. We have more ventral filaments that usually are anchoring the podosome actin tower towards the inner part of the cell membrane. And then we have about 500 nanometers inside the cell, these longer fibers that are actually much more connecting the podosome to each other. And now this will not probably surprise you, but the myosin 2 is mostly enriched at the dorsal actin fiber. So the higher you go in the cell, the more intense is the actin, the myosin 2A signal, and the less intense is the vinculin signal. It's not a problem of point spread function that is different for the different colors because we have swapped the colors. And I can tell you that here, if you do, again, a section, right, of normalized fluorescence intensity going from the center of a podosome at the very lower plane, then you see that there is much more vinculin enriched and almost hardly myosin. But if you go higher in the cell at this plane here, then you see a very high intensity of myosin all around the podosome core. And so then, and this is a zoom in, which I think it shows you very nicely how this myosin is really decorating these upper fibers inside the cell. But now what keeps myosin there, right? So why isn't myosin going down and start, changing all remodeling all the filaments? So then we started to think about septins, and this is work of actually it's ongoing work, unpublished work, which was interrupted by the COVID crisis, of course. And then septins, you might know, they are this cytoskeletal scaffold that are able to bind both actin and myosin. And then Koen van den Dries is a super talented postdoc who's now actually assistant professor with us. He started to look for septins, and we know from the work of Stefan Linder from Hamburg that actually there are 3 different septins present at this podosome area: septin 2, 7, and 11. And septins make this very heterogeneous network. They are very obscure, very attractive, very interesting group of proteins. And what we found is actually that the septin seems also to follow this higher level of organization of the actin, so the higher dorsal network. And the nice part is that if you stop, let's say if you block, inhibit the septin function, and you do this by transiently expressing this small peptide, which is actually known to cause septin aggregation, and so they sort of block the septin function, you do not disrupt podosomes, they will stay, but the peak of intensity of myosin, which used to be at this high level, if you go from the bottom to the top of a single podosome, I think you will see it nicely dropping. So somehow the myosin is not anymore on a very high level in this network, but much more on a lower level. Does this have any consequence on turnover of the podosomes? Well, actually it does. I think these movies are sort of mesmerizing if you really look at those. So in a normal disease, you will see this beautiful usual turnover of individual podosomes. But if you look at the BD3 expressing disease, this is actually LIFAC GFP signal, what immediately is clear is that they are smaller, they are there is some kind of static actin network underneath this cluster, which we didn't figure out yet. Why would that be? But the most striking thing is that the turnover of the podosome seems to be much faster. And so what we think is that by blocking the function of septins, myosin becomes more free to basically go from the dorsal part down towards the other filaments. And in this way, we'll start cross-linking and dissolving the podosome, which is actually known as a known function of myosin in podosome dissolution. of course, we need to understand whether there is any link with mechanical properties and what is actually the regulatory mechanism behind this. So this is also ongoing work that Kuhn is pursuing in the lab. So what happens now to this podosome actin architecture and dynamics if you put the cells on a real, let's say, a tissue-like compliant substrate? Because of course, super resolution needs glass, otherwise the quality is not very good. But I mean, so what happens if you put them on some kind of tissue-like situation? So what we did, actually, we put the dendritic cells on two different stiffnesses, very, very stiff, like in 600 kilopascal, and soft, which is about 2 kilopascal again. And this is PDMS, which is also pretty amenable for confocal, for microscopy in general. And there are three things that we noticed. The first thing is that there is this ventral vinculin-rich actin filaments of individual podosomes. There seems to be shorter on soft substrate. So meaning that if this is a cartoon representing the podosome, you have this inner part with the branched actin, then you have this alpha-actinin-rich sort of bundles, and then there is this vinculin here that is holding the structure to the membrane. If this is on the glass or on a stiff substrate, when you go to a soft substrate, you basically lose this. You don't lose anything else. You lose this component in here. The second thing that happens is that, and I think you will appreciate, hopefully in the quality of the movie, it comes across sufficiently well. You will appreciate that there is some kind of difference in the signal that you see from the vinculin, for instance. Now here you have a much more cloudy situation in between the different podosomes. And you have here a much cleaner and well-defined ring of vinculin. And what we lose the mesoscale coordination. So basically, if you also calculate the nearest neighbor distances of all these different features, then you see that they are much closer to each other, but they communicate for a shorter range. So whereas here, this podosome might be able to influence the movement of many more neighbors, here, let's say the mesoscale coordination is very, very proximal, and it doesn't go across many different, many micrometers. The third thing that happens is a functional implication. And it basically, podosomes or the dendritic cells will start degrading gelatin only when they really need to. So when the substrate is stiff, then the matrix metalloprotease machinery of the dendritic cells will be triggered somehow, and then they will start digesting. But if the tissue is soft, they will not do that, which is actually pretty logical, because these diseases are continuously in soft tissue in between other cells. So if they would start digesting things around, I don't think this is something you want to have on a physiological point of view. So what is that we want to summarize this is, I think we have identified some kind of actin modules that can sort of explain how podosomes sense the substrate at the nanoscale. So we think we have, and here is the model, right? The cartoon of the biologist. That would be really cool to have a real mechanical model. So we have identified a branched inner actin core, which we called the protruding core. This is the protruding module. This actin is polymerizing here and therefore pushing towards the membrane, only because there is this nice cage of bundle actin, which is nicely anchored to the membrane, which will also be polymerizing, of course, to formins. And then we'll give probably a counter force, and that's why the cells can push. At the same time, we have these vinculin-enriched filaments that are actually disappearing when the substrate is sufficiently compliant. They are not necessary anymore, so they are sort of reinforcing. It's a sort of mechanosensing module. And then we have what we call the connecting module, where we find myosin, and where we also we find septins. So now I think I can summarize this by saying that how substrate stiffness affects DC phenotype and functions. Well, I think we have seen that there are specific markers that are actually changing a little bit, and some cellular functions that are changing and are sensitive to this substrate stiffness. What the mechanisms are, which transcription factors are involved, are all very relevant questions that we will be very happy to address. And of course, we also try to understand how disease remodel the cytoskeleton. And I think the podosomes are selectively adapting actin architecture and producing forces in response to substrate stiffness. And because of the time, I'm not going to show you our findings on a 3D environment. I'm only showing you this, what I think it's a beautiful animation of 2 dendritic cells in a 3D collagen 1 gel, where you see again this beautiful nucleus deformation, the uptake of antigen, and of course this very nice fluffy actin arrangement. And understanding how dendritic cells in 3D works is basically an important part, certainly for the future. I want to conclude by telling you where are we going now. I would be very happy to hear your thoughts. And of course, if there is someone interested in collaborating, we're always happy to do this. We want to understand the 3D life of a dendritic cell, of an immature disease. It would be very interesting to understand what is the relation between this podosome remodeling and dendritic cell maturation. So what happens exactly when you have this massive amount of actin that becomes free suddenly when you add the stimuli? You can also imagine that this might be interesting for us to bring it up to a sort of studies for pathophysiological mechanobiology of dendritic cells. And we like to combine imaging techniques. In this case, we had a little study last year on a super resolution CLEM to investigate composition and ultrastructure of these structures. And I think this is where we are going at the moment. And I want to take one moment to thank the people involved in here. I already mentioned Koen, a very talented young scientist who did a lot on the nanoscale of podosomes. I want to thank Svenja Menens, who's a PhD student in the lab who started the mechanobiology of disease and the disease in 3D. She will graduate next week, so that's also very nice. Ben Joosten is a very talented technician who helps us with the imaging analysis. And of course, I want to thank all my collaborators, my nice biophysicists who help us with these different techniques. And of course, I want to thank you for your attention.
Thank you. Everyone, let's thank our speaker.
Thanks. There's a helicopter flying now here at the hospital. So
Timing.
I'm almost
Good.
there.
Then you can just assume there was thunderous.
Exactly.
Thank you so much. We have, I'm going to start us off with a question from YouTube where Vera Belavia has asked about a little more information for your substrate stiffness screen. So you concluded that there's this threshold at 12 kilopascals, but you know, have you thought about looking at other more intermediate values to see what's really triggering the integrin reduction around 12?
Yes. So we would like very much to look at intermediate thing. We have tried even softer gels. The softer gels are not really workable. And we have tried with PDMS to make PDMS ourselves and have a sort of gradient in stiffness. And we really see a gradient also in the degradation. And, you know, so it's really, really going down. But we did not try yet to go like, I don't know, 2 kilopascal above or below the 12 kilopascal. And that's something maybe we should be doing. There is a beautiful work of, well, actually Mike Sheets' lab, of course, when they are very much into this substrate. And then maybe we could get some inspiration in there for sure.
So we've got a question from Zoom from Sudha Kumari, who's asking why you think there are more podosomes after the septin disruption.
Yes, you know, this is a beautiful question. On the one, so I think it might have to do with myosin. I mean, you do need myosin to make podosome, and you do need myosin to break podosomes. It's a funny thing of myosin. You need myosin to make them. So could it be that by blocking the septins, you have more myosin available, and maybe this more myosin available is at a certain level helping to make more podosomes, but also because you have more myosin available, they will be also disrupted more quickly. But this is an excellent question, and Koen is very much looking into this right now, try to understand what does this come from, what is this, and also this funny there is an actin meshwork. If you really look underneath this podosome cluster, that seems almost paralyzed. So how is that possible? Why are there so many degrees of dynamic freedom in there? That's difficult.
Jen, you're muted.
Am I okay? Unmuted now?
Yes.
Oh, sorry. I was trying something new. It didn't work. So Janice Burkhart asks about the substrate degradation assay. Is it FTSE gelatin on top of the PDMS or glass? And doesn't that impact the compliance of the substrate?
So it is a very thin layer of gelatin, and the gelatin is an Alexa conjugated gelatin. And it should, because it is very thin with respect to the thickness of the entire PDMS, we don't expect to change the compliance of the substrate. We have tested the substrate compliance with AFM measurements. You know, this whole so we know that they are soft and stiff, but you know there is a lot of debate on which measurement you need to use to really establish the stiffness and softness. So it could be rheology, but then you're really looking at the torsion of the substrate and not really looking at what cells. So the AFM, we think, is a relatively good system to test the stiffness, the elasticity of the substrate, and we didn't see any important change.
Amrin Denane is saying, nice talk and movies, and has two questions. So the first is, do you think that there's a threshold force required to maintain the core ring structure, and are there any measurements? Do you want me to?
So like a threshold in size to maintain the core structure, that's the question?
I think he's nodding, so yeah, unless you want to come on and say yourself, Amrinda.
Yeah, hi, thank you. That's a very interesting talk. So I was just wondering if there are any force measurements or the values of force. You relate the experiments to the underlying substrate stiffness. But actually inside the cell is a force contractility. Does that maintain? And if you are below a certain threshold, do you have improper core ring structures or not fully developed structures?
Yeah, so we did not, very, very, very good question, very interesting question. So we did not look into that. We do know from recent work of the Salaida's lab and Olivier Desteng, that actually there are, of course, tensile forces in there that integrins are pulling on the substrate in the range of the piconewton. But we did not look into what you are asking. We don't know whether there is a threshold. We know that there is a maximum in the size of a podosome that can be reached. They are never larger than 500 nanometer unless you start knocking out some components, you know, and then you don't know anymore what you're looking at. And there is some minimum size. I mean, Benny Geiger has shown in the past really the starting of the formation of podosomes. We have tried to do something like this, but we always see also a very fast imaging rate. The components, they all come together, and very immediately you start seeing these different small substructures there. You immediately alpha-actinin is in a sort of donut, and even if the podosome is 200 nanometers at the most. So there's some kind of requirement to be called a podosome. It's not only a little dot. Yeah, I don't know if this answers your question, but we don't know more than this. That's a problem.
Can I ask my own question? Oh, Amrinda, do you have a second question? Sorry.
Yeah, my second question is, I think you alluded to the 3D environment. If in the 3D environment, you really don't have as many attachment points, for example, if it's a loose connective tissue or so, would you still expect the same spatial density and networks that are forming?
I'm happy you're asking this question. The answer is no, I would not expect that. So my personal, I mean, there are groups that have shown that these protrusions are formed at the end, for instance, of a large, if you have a large cell protrusion, these cells are very, very flexible, and they really squeeze in between all kinds of fibers. And there is nice work of different labs showing that at the end of this larger protrusion, you can find some dots that are unreached in the podosome markers. But obviously, as you also point out, this is not the same, right? It's not the surface. So my personal opinion is that we cannot expect podosomes like the ones I showed you in a 3D collagen gel. what I would expect, and people have found this, is that the moment that the dendritic cells will touch what it can be a 2D surface, which is present in our body, then there you have more the formation of these clusters. The most extreme example is, of course, the osteoclasts. They make on the bone surface, they make this enormous, gigantic podosome-based structure. And so, no, in the 3D, I'm not going to fish for this protrusion, but I have the feeling, and I think also from the work of Isabelle Maridonneau-Parini, that the ingredients are the same. They are just being used in a different way. So you have the proteases again, and you have, of course, the integrins again. So it's just a little bit different. But of course, we know also that they use integrin-independent migration, and also work from Michael Six. It also showed this. So they're very plastic cells. The integrins, in my view, are used only in the moment that they really have to stick to some surface, and there is some ligand in there.
Thank you.
Well, thank you.
Quick question from me next, which is related to how stiffness in the in vivo environment might be involved in the guidance or movement of the dendritic cells and the podosome relation there.
Yeah, you know, I'm gonna share with you my secret dream. So I would be very interested in understanding whether dendritic cells that encounter highly fibrotic tissue, so a very dense and very stiff tissue. It could also be a scar-like or, you know, the tumor microenvironment. Whether those signals are responsible for keeping the dendritic cells in an immature state. And if they are in an immature state, even if you give immunotherapy, you know, even if you give signals, they will not be able to migrate to the lymph nodes. So it is really intriguing, I think, to understand this component in the dendritic cell immunobiology. Very challenging, but I think that's exactly the point. I think we have evidence from previous work from our lab that if you put the dendritic cells on these topographical cues, you know, and then all these podosomes nicely align as if you are, I don't know, making a huge collagen bundle. And then we give the prostaglandin E2 signal, that the podosome do not dissolve, and the dendritic cells remain much more in an immature state than on a flat surface. So somehow the engagement of the integrin in this way seems to prevent, but it's not a black and white situation.
Yeah, thanks.
Yeah, thank you.
Hi, sorry, I had to find my mute button again. So Mater asks, it was my impression that the podosomes are not distributed evenly at the base of the cells. Do you know what organelle, maybe the nucleus, is on top of the podosome-rich areas?
Right, that's also a very, very sharp observation. The answer is podosomes can be formed in different aggregations, in different cell types. So in osteoclasts, they make like a belt all around the cell, like a large ring. In dendritic cells, they usually are in clusters that are much more at the periphery of the cell. We know the works showing that invadopodia underneath the nucleus, they get this piston-like force, and then the nucleus on top of this invadopodia is actually helping the invadopodia to push down. Our podosomes are hardly underneath the nucleus, mostly at the periphery in very thin surfaces. So it could much more be that the upper membrane of the cell is actually laying on top of the podosome clusters rather than a nucleus. We know there are mitochondria that can be found in between podosomes. We have never found any other organelle associated really in a sort of stable manner.
Okay, we just go over to YouTube now. So a question from Madhurima Sakar. She said, hi, very good talk. I am just curious, did you check stiffness dependent changes in macrophages?
I did not, but I know that there are people looking into this, and they seems to be sensitive to. Yeah, you should be probably contact. I always contact Stefan Linde, or I don't know, something about macrophage podosomes. That would be is the guy to call.
I mean, really, who isn't mechanosensitive? Another question from YouTube from Christina Bertocci. So can we say that this is another example of vinculin acting as a molecular clutch for the differential engagement of the actin-myosin modules, similar to what happens in adherens, junctions, and focal adhesions?
I would be tempted to say yes. Well, we know that there is vinculin and that vinculin is under tension. We have used this vinculin tension sensor of Martin Schwartz's lab, and we do see in a Fred Flynn dependent manner, it's a previous work a couple of years ago, that we can see changes in the lifetime. So vinculin is under tension. So I would imagine that that's, the ingredients are there. Yes.
Okay, awesome. And then I think this will be the last question. Emily Richardson has one that was already sort of addressed by Emrinders, but if you can talk a little bit more about it. So when you look at the cells in a softer 3D environment, do they tend to migrate in a more amoeboid fashion than in the stiffer 3D environment?
So the 3D environments that we have used is our collagen, fibrillar collagen 1 gels. And I don't know whether we can really say that they are softer or stiffer. We have made a different density of collagen. So I'm not sure we can say the difference in that. We like in that case to talk about density, not so much about stiffness. We didn't even measure that, so I'm not going to say anything about that. But what we see is that in the low collagen density, of course, the diseases are much more free to move around. In the, let's say, more dense collagen gels, which is we have observed in a couple of donors that you see the disease that makes up like little tunnels. They go back and forth. They seems to degrade when they cannot really do anything. We see some pericellular collagen degradation, which is MMP dependent, because if you block it, then you don't see it anymore. But other than that, we have not looked into this, not so much into the stiffness. But it would be interesting to do this, maybe by cross-linking collagen or using some tunable polymers that make 3D gels. That would be also really nice.
Okay, great. Well, I think that was a great last question. So I think we're pretty much all set. So thank you again to both Adrian and Alessandra. It was really wonderful having both of you today. And I think this will conclude seminar number 8. Thanks, everybody.
Thank you.
Thank you very much.